Well, thank you. Thank you for the nice introduction. Yes, it's a pleasure for me to be here and to tell you a little bit about the road that I've traveled and trying to understand what single nucleotide variation is actually doing in our genomes. And at the end, I don't have a question for you. I don't have a complete answer for you, but yeah, maybe a little contribution. So I've understood that all of us here are population geneticists or population genomicists. So I guess you more or less have an idea of how the field of genetics has evolved. So it started in the end of the 19th century when Gregor Mendel came up with the idea of Mendelian inheritance and how different variants can be inherited from parents to offspring. Well, these ideas were further developed at the beginning of the 20th century, for example, by Thomas Hunt Morgan, one of the famous Drosophilists. And then, of course, the structure of DNA was discovered. So this is all at the beginning of the 20th century, and this was all done. And it's only fairly recently that we also have been able to sequence DNA. And for many of us, this is now an important part of our work. So looking at DNA sequence or RNA sequence and trying to figure out what it means. And we're now in the age of the omics revolution, I would say. So since the end of the 20th century, large genome sequencing projects have been possible. And this has basically given us an unprecedented volume and resolution of genetic data. And because of this, and also because the costs have dropped well considerably in recent years, omics data is now within reach for many different research questions, but also for applications. Well, some clear examples you can see, well, even at home. For example, if you go online, you can have your own DNA sequenced. So this is not a whole genome sequencing, this is marker-based, but you can pay a couple €100, at least that's how much it cost me a couple years ago. You take a swab from your cheek, you send it in, you never even see these people, you pay the bill, and then some weeks later you get a report back telling you everything about yourself. So one of the things that this company told me about me was that I had 80% more Neanderthal DNA than 80% of their customers, which I thought was very useful information. So you can see it's about 2%, which is the fairly average number for someone of European descent. But they also gave a lot of other information. So they gave me reports on all sorts of different traits and telling me based on my genome sequence what they predicted my traits would be. So whether I would like sweet versus salty. So they said, you likely prefer sweet. Well, this is true. My longer toe is indeed longer than my other toes. I don't have a unibrow as far as I know. Also not secretly, this is just how it is. And my wake up time, it said, was 8:34 p.m. or a.m., which I think is a really nice wake up time. It really suits me, but unfortunately, it does not fit the schedule of the kids and a job an hour commuting away. But I think this is pretty amazing. So all this by just sending in a swab with a little bit of my DNA. Of course, they also came up with more serious reports, giving me prediction scores for all sorts of diseases and conditions. So then it gets a little bit more serious. And it makes me wonder, how reliable is this? How well can we actually link genotype to phenotype? And this is a question I find very interesting. And this is basically a question that has driven my research since I became a postdoc at Lausanne. So if we think about the genetic code, about DNA, you can say it's basically a cookbook for phenotypic variation. And there's many different ways of looking at it. And this makes it complex, but also very interesting. And which of these aspects of the recipe are most critical? That is one of the key questions that we want to solve. Should we look at individual letters in this recipe? So sort of like the nucleotide variation. If apple becomes appy, it's a bit different, but it's still quite similar. salt. If it becomes malt, then it's quite a different ingredient, so maybe then it does matter. It depends. Or should we look bigger? Should we not focus on these little typos, but try to think like, okay, what do the words mean, and what does it mean to the recipe? So let's say at the gene level, if you replace an apple with a pear, you get a different kind of cake, but it would still be quite edible, I would say. If you replace apple with broccoli, well, then it's a different story. And then, of course, you can also look at quantities. So 1 tablespoon of salt is not exactly the same as 10 tablespoons of salt for an apple pie. So that will be looking at gene expression. So there's many different ways to look at the genetic code and trying to interpret it. And what I've been doing is mostly focusing on the nucleotide variation. And this is what I want to tell you about today. So linking genotype to phenotype is not easy. And we could talk about it for hours, why it's not easy. I just want to highlight a few things to keep in mind. So if we think about genotype, so about DNA sequence, over, where's the point going? The pointer's here, over here. And on the other side, looking at your phenotype, which could be, well, could be anything. It could be fitness related. There you can see that there's many different levels in between there, and many different steps that the information takes. And often going from one step to the next level and then to the next until the final phenotype, it's not very linear. There's many different models describing this. I'm not an expert on these models, but it's often not very linear. It's not very easy to say like, okay, we have this DNA sequence, so we will have this RNA sequence. There's complications in translating from one step to the next. And of course, there can be influence of all sorts of environmental factors. So another problem is that if we think about genetic variants, there's just so many of them that it can be daunting to just start investigating it. If we think about SNPs, so single nucleotide polymorphisms, it has been described that over 600 million SNPs have been recorded in human populations. For fruit flies, for example, which is my model organism, it's a bit less, but it's more than 4 million as well. So still large numbers. And what you see is that if you take different populations with different genetic backgrounds and they evolve towards a certain phenotype, that they can take very different genetic routes getting there. So there's a certain level of redundancy for many phenotypes that we look at. And what can help in trying to find your way around so many of different SNP variants are all sorts of genome scans. So you can do a GWAS analysis, genome-wide association, trying to associate your phenotype with specific variants, approaches such as evolve and resequence. And this can help narrow down your list of SNPs that you want to look at. But still, there tend to be hundreds or thousands of different SNPs in your candidate list, which is still quite a lot. And the problem with all these different SNPs is that they tend not to be independent from each other. And you can also not consider them to be independent. Because one thing that is very important is genetic linkage. So if you have variants that are related, located closely together on the genome, they can be genetically linked. And because of this, you can have genetic hitchhiking. So this is when you have a certain variant that is under selection, the red one over here, and then there's some other variant that is, well, doing who knows what. And if this red one is then selected for, it will increase in frequency in the population. And the other one is linked and it will just be dragged along. So what you get in your lists are probably certain variants that are under selection and other things that might not be under selection. But really, we don't know very well what different variants in candidate lists are doing. Well, and to make it even more complex, genetic background can have an effect as well, through epistasis, for example. we didn't even get to this in my line of research, but this is also a complicating factor. So the key message is it's not so easy to link genotype to phenotype, but we want to try anyway. And we think that Drosophila melanogaster is a pretty good model to do so. And we think this because it's a powerful genetic model. So there's all sorts of genetic tools, transgenes, genome data sets. They're also fairly easy to rear and to maintain in the lab. And what we see is from this list over here is that research on Drosophila has contributed to our understanding of the mechanisms of a lot of different important phenotypes and traits, hence being awarded a bunch of Nobel Prizes in the past. At the same time, and well, I'm an evolutionary biologist, or at least that's how I identify myself, Drosophila is interesting because it also allows you to ask evolutionary questions. So there's a lot of populations. Drosophila melanogaster is a cosmopolitan species, and local populations have adapted to different conditions. So there's a lot of natural variation. This is a figure from a recent study that is currently under revision, but it is, I think it's available as preprint. I'm not entirely sure. And what we see here is that through large consortia, collecting populations from different locations and sequencing them, we know quite a bit about genetic differentiation of populations of Drosophila. But what you can also do with melanogaster is experimental evolution. So this is also a very handy tool for asking experimentally or evolutionary questions. So altogether, this package provides unique opportunities to study genotype to phenotype connections. And in my group, we have focused mostly on life history evolution. So we look at the life cycle of Drosophila, and we look at variation in lifespan, fertility, but also other life history traits. And we consider this interesting phenotypes because they're closely linked to Darwinian fitness. They're complex polygenic traits. At some point, you may, well, I've often thought that maybe it's a bit too complex. I should have maybe picked an easier phenotype, but yeah, we kind of think this is interesting, so we go for it. And what we also think is interesting is that there's all these life history correlations that we see in natural populations, but also in experimentally evolved populations. And the question is then, how does this work genetically? Can we try to piece this apart a little bit and say something about it? So throughout my career, initially I started out with just trying to elucidate the genetic basis of aging and life history evolution. This is basically the first grant I got as a postdoc was about this. And then gradually, I became more and more interested in understanding specifically what individual SNPs are doing. And I will show you the work, starting from my postdoc work up to the recent work that my own PhD student is doing now, to see what kind of paths we took and what are the lessons that we learned. So we started out, I didn't start out making experimental evolution line. I just hopped into the project when the lines were already established. But basically, what we started working with was with a set of lines that had been experimentally evolved to study aging and life history. Well, in Drosophila, this has been done several times. One of the selection regimes that is often used is selection on postponed reproduction, and this reliably gives a response in longevity as well. And another way to play around with longevity and aging is to play around with diet or starvation resistance. So the study I worked on, we actually mixed the two components. So we did a multifactorial design in which on one hand, we had 3 different developmental diets. So this is our control diet. So this is the normal amount of yeast and sugar that we have in our fly food. And then there was one treatment with a reduced amount of yeast and sugar and a treatment with an increased amount of yeast and sugar. And this was during development. During the adult stage, they were all on control food. And then we combined this with this commonly used early versus postponed reproduction regime that has been applied several times already in the past, because we thought, okay, this is going to give us an effect on longevity for sure. Well, this experiment had been going on for almost 60 generations for the postponed reproduction lines, and almost 120 generations for the early reproducers when I started with this. And this was the point that we thought, like, okay, people have already phenotyped these lines. We know that there are phenotypic differences. And now we also want to see what kind of genetic differences do we see. And we used PoolSeq to do this. So we sequenced all these lines. And then the first thing that we did was to just create an overview of genetic differentiation among these populations. And what we see is, yes, indeed, these lines differ phenotypically and also genetically. So the late reproducers, which are all in the circle below, we see that these have a higher lifespan, they live longer, they are bigger, and they reproduce later compared to the early reproducers. Then we also see effects of diet, especially of the poor diet. So the control and the high diet regimes didn't differ so much from each other. And this regime had especially effects on development time and adult size. And then this was a bit of an outlier group. So this is the combination of the low diet and the early reproduction. This had a particularly strong effect on development time, but also a little bit on lifespan, on adult size, and early fecundity. And we feel that this has been the most stringent regime of all 6 combinations, because it basically forced them to develop as fast as they can on this poor diet and then switch as quick as they could to reproduction. So this is a general pattern, but what we really want to do, what I wanted to do, was to identify genome-wide associations between genotype and phenotype. So where in the genome is this happening, this adaptation? So as I said, we did a whole genome sequencing, so PoolSeq, of the experimentally evolved lines. And I was particularly interested in the contrast between the early and the late reproducers because, well, I got a grant to work on longevity. People want to fund research on aging and longevity, so that's what I worked on. And what we found was almost 400 differentiated SNPs in 150 genes. This is great. This is a nice data set, but it's still quite a bit. And if we look at this picture, this is like a typical Manhattan plot in which you see all the SNPs that are in the genome. And these peaks, they represent, I don't even know where my pointer is. Oh, there it is. I'm too fast. And these peaks basically represent how differentiated the allele frequency is between the early reproducers and the postponed reproducers. So the higher, the more different. And the red ones are the significant ones then. And you can see that these significant SNPs, they're not randomly distributed, but they're located in clusters. Some clusters are fairly big, such as this one. Some peaks are a bit more narrow. But this raises the question, like, okay, what is going on there? There seem to be multiple regions that are involved. We expected this to be a polygenic trait, but then what in these peaks is really interesting? There's probably some form of genetic hitchhiking. Should we just consider each peak as a haploblock, or can we say something about what is in these blocks? Well, I decided to try to look what was in the blocks. So we annotated all these SNPs. We looked up which genes they are, did enrichment analysis. Well, nothing was functionally enriched. So then I just looked at all the genes, and I looked at the literature, and I tried to pick out a couple that seemed to have, well, at least some hint that they might be involved in life history evolution. And the one gene that I worked on most is the gene ecdysone-induced protein 75B. And this is purely because it was in the list and because from literature we already had an idea that this might be an interesting one. So this is a gene with broad effects on life history traits. So the effects on metabolism were already described, for example, but also effects on stress resistance. And what I first did was to see, like, okay, if we play around with these genes, if we manipulate it, so we knock it down using RNAi, do we then also see an effect on the phenotypes that we're interested in, so longevity and fecundity? And then we also measured egg volume because, well, we had eggs anyway. And in this case, in this study, I used an inducible transgenic system, and using RU, this is a drug that you can use to induce RNAi. So when you use your RU drug, when you feed that, you turn on the system, and then you downregulate your gene, and then we saw effects on longevity, but we also saw effects on fecundity and egg volume. So this was good. So this was encouraging because we found a gene, and then it turns out it can also actually play a role in the phenotypes that we're interested in. The big question is then, of course, are functional tests at the gene level representative for what candidate SNPs are doing? And this is a question we couldn't answer with just RNAi. So if we look at different mutants of a single gene in Drosophila, we know that different mutants of the same gene can be quite different from each other. So different SNPs, they can also potentially have different functions. And how do these gene level tests or the RNAi then tell us something about SNP function? Well, we concluded that it really didn't do it. So I did allelic replacement of individual SNPs using CRISPR-Cas9. Well, one of them worked out well. So basically what you did, and it looks very simple, put like this, you take a strain with a certain sequence, you take your target SNP, and then you replace it with the alternative. And then you have two lines that are, well, as identical as they can be, given practical limitations, but that differ at this SNP, and then you can test that. And this took a bit of work, but it worked in the end. And I specifically worked on this SNP, which was one of the SNPs with a pretty high p-value, not the highest actually. And the results, they made me very happy. So what we saw here is that if you have a T and then you replace it with a G, is that the lines that we got had a higher fecundity, so more eggs laid per female. And this was a pretty strong effect of about 15%. So for a single SNP, I was quite impressed. So we also looked at longevity, which was the other trait that we were after. And here we see some variation, but nothing significantly different between the lines. And since we had the line, we just phenotyped the hell out of it. We also looked at egg volume. There was no effect. But then if we looked at egg to adult viability, there was a strong effect as well. And a couple of other phenotypes we also didn't see an effect. So this was quite exciting. So I found a SNP, we CRISPRed it, and we found very, well, I would say strong effects on fecundity and viability. So it made sense to why these SNPs were there. They might have been under selection and explaining part of the response in fecundity in the experimentally evolved lines. There was no effect on longevity and other life history traits, but this could be other loci that are doing this, of course. And this basically tells you that when you do gene level tests, when you do RNAi, when you work with mutants, it is not really representative of SNP function. And if you really want to know what a SNP is doing, you should go and try and look at this SNP. And this is not easy, but if it works, it can give you some interesting results. So how to go from there? So we had an open question here, and this was, okay, we have now a fecundity SNP, but perhaps this gene is also involved in the longevity differences. But then maybe it's another SNP. So we think that functional tests at the level of SNPs are critical to understand life history evolution. But then where do you start? So we did one. I think we got pretty lucky with it. That was a lot of work, but we got nice results. But then within E75, there's depending a bit on your statistical cutoffs, there's a couple of significant SNPs up to 16, which is quite a lot for functional tests. If you want to extend your search to all the other SNPs that we detected in our study, this will be even more, of course. So where do you start? Because knowing how much work it takes to study a single SNP, you'd better pick wisely what you want to do. So I felt when I finished my postdoc and when I started as assistant professor at the VU Amsterdam, that we needed a better understanding of patterns of functional impact among SNPs. And if CRISPR would work very easily and very efficiently, then you could CRISPR the hell out of it. But right now, this is not the case. So this is why my PhD student, Julia, took a different route to study this problem. And she's making a tool, or she has made a tool, to predict functional impact of SNPs. And we call it FlyCat. And basically, the idea behind this prediction tool is that the position of a nucleotide in the genome will provide information about its function. Because the position can tell you, for example, whether a SNP is located in a coding region or a non-coding region, whether it's in a repetitive region, a regulatory motif, is it an evolutionary conserved region, well, etc., etc. There's many different annotations that tell you something about a nucleotide position. And the idea is that you can use these annotations to predict the functional impact of SNPs as an additional method to trying to understand SNP functional patterns. So the approach that Julia took was that she basically followed the combined annotation-dependent depletion framework, which is CUT in short. And this is not a term that we came up with ourselves. This was first developed for humans, and they came up with this term. But basically what this tool is, what this framework is that it's going to combine different annotations into a single score for phenotypic impact. And it makes use of a machine learning algorithm and the functional impact prediction score, and it will provide a functional impact prediction score for all possible genomic variants. So not just for variants that we know exist in population, but basically for every single position in the genome. So the first cut was developed for humans, but since then it has also been developed for mouse, for chicken, and for pig. But so far, not for any insect species. And this is where Julia's project comes in, because she developed a FlyCat specifically for Drosophila melanogaster. And this is possible because there's a lot of annotations available for this species. Well, this is just a screenshot from the UCSC genome browser that shows already some annotations, but there's a lot of databases with a lot of information, also on evolutionary conservation, on epigenetic data, regulatory regions, and all of this can be used to make a FlyCat. And she actually succeeded. The preprint came out last week. So if anyone is interested to learn more after the presentation, you can go have a look and read it for yourself. But what I want to do is to briefly explain how does CUT work, how does FlyCat work, what are the assumptions it was built up, and how can you actually use it. So the leading principle of FlyCat, or of any CUT, is that you need a multi-species alignment. Well, luckily, there's a lot of genomic information available for Drosophila species. Over here we have melanogaster, but all these other species have been sequenced recently as well. And what this does, it allows you to reconstruct the ancestral sequence. And this is important because FlyCat, or any CUT, works under the assumption that derived variants that we only see in melanogaster, but not in the ancestor, tend to be neutral or benign, and that over evolutionary time, high impact negative variants will be depleted. So basically, how evolutionary theory works. So we reconstructed the ancestral sequence of this particular ancestor over here. And then we compared this sequence to the sequence of modern Drosophila melanogaster, assuming that since this ancestor there has been purifying selection that has depleted variants with a high negative impact from the lineage. So variants that are absent in the ancestors, but that are nearly fixed in modern Drosophila melanogaster sequence are more likely to be either neutral or to be benign. So this is basically your one set. So the other set is taking this sequence, so the reconstructed ancestral sequence, and then simulating what could happen if you would evolve to the same point as Drosophila melanogaster is today. So you basically simulate the data set in which there was no purifying selection, because this was not in our simulation, but just creating genetic variation because of mutation. So this simulated data set is more likely to have variants there with a high negative impact. So these are your second set. And what FlyCat is doing, what the algorithm is going to do, is that it's basically going to compare these two sets. So the neutral benign variants and the simulated variants. And it's going to look at different annotations that we have. And it's going to learn which annotations are associated more with one set or with the other set. So we had 38 different annotations, and we also made combinations of different annotations, resulting in 691 features that the model used. And this was done for almost all chromosomes, except for the Y chromosome, because there was too little training data available. And the machine learning algorithm then basically went through all this data and through all these annotations and came up with a weight for each of these annotations on how important this is. And once you have this, you can give it the reference sequence of Drosophila melanogaster, and then it will calculate a score for each position in the genome based on this. So we have now this list that has a FlyCat score for each variant, and it is basically a score that is scaled between 0 and 1, with Han having the highest predicted phenotypic impact. And it looks something like this. So we have the reference sequence, and then for each position in the genome, you will have 3 different cut scores, so a G to an A. Substitution would have a score, for example, of 0.6. If G to C would 0.63, and then G to T would be 0.61. And then if you look at the different position, these scores may be lower or higher. So this is basically what it is. So what we have is a list of all the positions that is available for use, and that people can look at the scores for their favorite positions. And of course, well, I'm really not so much of a modeling person, so I'm always a bit wary to trust what comes out of it. But Julia did all sorts of tests to see, like, okay, does it make sense what kind of scores this FlyCat is producing? And we looked at the average, for example, at the average impact scores according to what we would expect biologically to be relevant. So these are protein level predictions, what we call them in the model. So basically annotations telling whether a SNP position is located in, for example, an intron or an exon or intergenic. And then what you see here in the red box is that if you look at non-synonymous mutations, is that they tend to have a pretty high CAT score. So this is what you would expect, because these are the kind of mutations that will affect the protein structure. And of course, there is variation. Not every non-synonymous mutation will have the same phenotypic impact. But this is basically the kind of scores you get for these types of SNPs. And over here, you see the scores for non-synonymous SNPs or for synonymous SNPs. And you can see that on average, they are lower, but there is a large variation. So some of the synonymous SNPs are predicted to have an impact, even though they are synonymous. I'm sorry? No, there was not enough data on polymorphisms, actual data to use in this evaluation. Yes. Yeah. Yeah, so this is basically what CUT is doing. It's giving you a score for every position in the genome, whether or not it exists or not. So if you, in the future, you have the genome and you want to make a mutation that doesn't occur in populations, then you can do it, and you can look at CUT first to see if you would expect impact. Yeah. So what is important is that CUT doesn't only work on a coding region, but it also works for non-coding region. So what you see here in the green box is, for example, the average scores, the score distribution for intergenic region and for intronic region. And you can see that it's all over the place. So some nucleotides that are in intronic or intergenic regions have a very high predicted impact. And this is interesting because this tends to be overlooked. So when we get our data back, we first tend to go to see like, okay, which of these SNPs are in coding region, and are there any non-synonymous ones? And then, well, if they're not, then you're going to look at your intronic SNPs, but then, yeah, you don't really know what to look at. So this is an example of a gene. This is the white gene, actually, I believe. Yes. And these are all the CUT scores in this gene. So what we see here in the slightly darker gray regions are the exons. And we can see that there's quite high scores on average, but there is variation. So this is what you would expect. There's a lot of potential synonymous, non-synonymous mutations in there. But what is interesting is that if you look at this large intronic region, is that you also see a lot of variation in predicted impact. So some of these regions have seem to be like almost peaks, where there is a higher predicted impact on average. Others tend to have a lower predicted impact. And in some cases, if you look at the data, you can say like, oh yeah, this is where a known regulatory region is. But for other regions, yeah, we don't really know, but then this prediction score is there. So this gives you a new insights that might be valuable to work with your list of SNPs. So all of the pictures I've shown you so far, and what we've talked about, is basically all the potential SNPs in the genome. Well, not all SNPs in the genome that can potentially exist actually exist. So we have over 4 million known SNPs in Drosophila populations. And if we look specifically at the known natural variants, and we look at their allele frequency distribution, then it looks something like this. So what we have here is, depending on allele frequency in natural populations, we plotted the flycat score distribution. But you can see that the median tends to be around 0.04. And of course, there are some high scoring variants. So they are tolerated in natural populations, but the most of them tend to have like intermediate scores. So this suggests that a functional impact, a predicted functional impact of 0.4 or 0.5 is normally distributed in natural populations. And this gives you some idea like, okay, what kind of scores would be relevant if you're going to use flycat in your studies. So how to use it? So what we anticipated, or what we wanted to use flycat for ourselves, was to distinguish causal variants from neutral variants. So we have lists of genes, or rather we have lists of SNPs that are too big to all investigate. And can we somehow try to tease out which might be causal variants and which might be neutral variants. So the pre-computed scores are all available, and we have Julia did a couple of case studies to show how you can use a tool such as FlyCat. First of all, to interpret GWAS studies and to also prioritize using choosing SNPs for functional studies. So the first thing that I'm showing you here is data from a study by Katzenberger et al. And this is a study in which they used DGRP lines, so this is the panel of inbred lines, to study death after traumatic brain injury. They came up with a list of SNPs, and what you see here is that if you look at the p-value, they figured the ones with the lowest p-value were most interesting, so they are highlighted in purple here, and then there was also plenty of others with a pretty high p-value. But if we then look at the cut scores for all these different variants, you see that there's not really a correlation between the two. It's basically all over the place. So something that has a high p-value doesn't necessarily have a high cut score. And this suggests that p-values, I mean, they're of course very valuable in working, trying to make sense of your GWAS. But if you then want to take the next step and try to see, like, okay, which of your SNPs are most interesting or potentially functionally important, it's good to have a second tool to also help predict this. So in this case, this study did not do any functional validation, so we cannot see, like, okay, do our cut scores make any sense? But this is the case for well, a few other studies, and I want to highlight this one. And this is a study on pigmentation. It's actually from this group. So I'm honored to tell you about this data. And what they found out is that they looked at different populations. So populations differ naturally in pigmentation, and this is also associated with lifespan and fecundity, among others. And also, if you do experimental evolution, you can see that you can readily evolve this. And then if you do genomic analysis of this, Bastida et al. Came up with 17 SNPs associated with a natural variation in pigmentation. So this is a nice number. This is a workable number. And a study that came after this, they actually did functional tests on a couple of these SNPs. And this is really exciting. I think everyone that does functional tests on SNPs should be applauded. And they focused specifically on 3 SNPs located in the cis-regulatory region of TAN. And TAN is a gene that could have an effect on pigmentation, but it's a pleiotropic SNP or a pleiotropic gene, so it could also have other effects. And when they did these functional tests, they did a really nice, they had a nice setup. They didn't look just independently at each SNP, but also at the combination. And there were a lot of epistatic effects, but overall, you could also say like 2 of these SNPs have effects as predicted, so they affect pigmentation. And one of them was a bit weird. It seemed to have a weaker effect and then going in the opposite direction. So if we look at these SNPs, so in blue are again all the potential SNPs in this part of the genome, so also positions that are not polymorphic in natural populations. And then over here, the 3 dots are the ones that were studied by this group. And the 2 that had an effect in the predicted direction on pigmentation have a cut score around 0.5, which is an average cut score. But we think that this is a moderate, this is a number that is worth investigating in most situations. And the one that was doing behaving a bit differently than expected actually had a really low cut score. So this is interesting because there was some phenotypic effect, but not as expected, and the cut score is also very low. So we don't really know based on 3 SNPs what that means. Ideally, we would do functional tests on way more SNPs, get more data sets to try and validate how CUT works exactly and what kind of scores produce meaningful phenotypic effects. So 3 other SNPs that were not investigated functionally are located over here, so a little bit further on the X chromosome. And if you look at the CUT scores, you can see that one of them has a pretty high CUT score of about 0.7. So if people would want to do more studies on this, then I would say like, okay, maybe go for that one. That might be functionally interesting. And this is a way how you could use CUT. So you don't have to blindly pick a couple of SNPs and put years of work in it, but maybe you can make a more informed guess. So another kind of application for CUT, which is not necessarily evolutionary interesting, but it is useful from an application point of view, would be that it can help refine genome editing experiments. And the way we envision this is that if you do a CRISPR experiment, you design all your vectors to target a specific region, so your SNP of interest. And what is a common thing with genome editing experiments is that there can be off-target effects. So these can be random, anywhere in the genome. Sometimes they're predicted already by the software tools. But in some cases, people also make intended mutations. If you do CRISPR, then there's a specific recognition site that the enzyme will recognize. And if you don't destroy that site, then the enzyme will keep breaking the DNA over and over again. And this is a problem for CRISPR efficiency. So this is why people often not only induce their intended mutation, but they also induce a mutation in this this region, this PUM region, to try and make their CRISPR more efficient. But what do these? What might be the phenotypic effects of these off-target or intended off-target mutations? So one study that Julia looked at was a study on the gene NTK. It's involved in pathogen defense. And in this study, they wanted to do CRISPR on a specific mutation that they thought was interesting. And that's the one over here. And what this group did in their study is that they induced this mutation using CRISPR. And as you can see, it has a CUT score of 0.47, so that is pretty nice. And then they also induced 3 other mutations nearby to help in the efficiency of the CRISPR procedure. And one of these induced mutations had a low impact score. The other was also fairly low, but one of them had an impact score that was actually about the same as the target mutation. So the target mutation was a non-synonymous mutation. So they expected like, okay, if we're going to target this, then we're going to see phenotypic effects, and then that's it. Then this is the reason. And these off-target mutations, they were all synonymous. So they expected them to be silent. But based on the CUT score, maybe this mutation over here might have an effect. And this might interfere with the phenotypic measurements. Well, next to this, these intended mutations, these authors, I think they did a really good job. They also analyzed the genomes of their lines to see what kind of off-target effects there were. And they found a couple, including one that has a CUT score of 0.99, so which we would predict to have almost certainly a pretty large phenotypic effect. So here you see that, okay, this might have an effect on the phenotypic measurements in this study. And if you know this, maybe you can design your study differently. So for example, don't induce this mutation, but try to stick to mutations with a really low CUT score. Yeah, off-target effects you cannot fully escape always. but knowing that they're there and that they might have an effect would already be important. So basically, this is what flycat is. And it has been possible to make flycat because for Drosophila melanogaster, there's all these high quality genome sequence data and annotations available. And what we do is that, what it does is that it allows for a better nucleotide resolution analysis of functional impact. And it doesn't solve all the questions. At the beginning of this presentation, we talked about difficulties of linking genotype to phenotype. Well, CAT is not solving everything, but I think it does contribute to, well, hopefully better understand and better facilitate studies on SNPs. And what is important, and this is something that not many tools do, is that it provides impact scores for both coding and non-coding sequence. And we know that non-coding sequence are very important for many evolutionary interesting processes that we as evolutionary biologists are interested in. So, as I mentioned, the paper is out. You can look it up. So to wrap up the seminar altogether, basically I've gone from looking at a phenotype that I thought was interesting to going more to understanding what SNPs are doing. And even though it's not easy, I argue that we need a better understanding of the functional impact of individual SNPs. And this would help improve our understanding from genotype to phenotype connection. So if you have a group of genes or a block of SNPs, so a haploblock, what are these SNPs doing there? Do they all have a little impact? Do some have a big impact? Do they all have effects on different phenotypes? Right now, we know very little about it. And then, of course, there are still problems like epistasis, linkage that cannot easily be broken, but let's just take it step by step. And I think this can make a useful contribution. And I think that we should do this by combining as many of different tools as we can. So genome sequence analysis, use GWAS in smart ways or evolve and resequence, but also try to do functional genetic analysis on SNPs. So this can be specific SNPs, but maybe we can also do random SNPs and just try to build our understanding of what different SNPs do. And then the newest tool to the toolbox that my student Julia made is that hopefully the functional prediction that flycat provides can add something to our understanding. And with this, I will have to thank a couple of people and funding agencies. So this project started a long time ago in 2014 when I came first to Lausanne. And the work there was done in collaboration with the Bas Swan group, Thomas Vlad group, and Laurent Keller's group. And the newer work, so the work on the flycat and ongoing CRISPR work, is being done at the Amsterdam Institute of Life and Environment, who generously supported Julia's project. And it's not just me, but I especially want to highlight Professor Meerte Bosse. So she's the person that has developed CAT for chicken and for pig, and she's been well, valuable, super valuable for this project as well. And thank you to all these institutes and funding agencies that provided money and facilities along the way. And thank you all for listening.